Hello, it's 8 o'clock in the evening and we are starting this training, this presentation. This evening we've got an honor guest with us, who is Professor Dr. Peter Dartsch from Dartsch Scientific. And after this fabulous training on alpha and beta, he will now present a different product. And this product has been highly praised on social media. We've got many testimonials. And now we are quite curious to see what the presentation will tell us in order to defend this product. And I would like to invite our guest of honor who will present that product, QUANT ARQ. Professor Dr. Dartsch, it's your turn. Please go ahead and welcome. Hello, Jérôme. Good evening, ladies and gentlemen. I'm really happy 1 week later at the same venue to be back with you and to tell you most fabulous things about a wristband called QUANT ARQ. We have tested the biocompatibility of that product and its effects in oxidative stress. Same as alpha and beta essence, we work with organ-specific cells, and I will talk about that in more detail later on. First of all, let me briefly introduce myself and the company, the research institute for which I work as director. I'm a graduate biochemist and I'm habilitated in human physiology. I'm a professor for human physiology at the University of Tübingen. I'm retired now, but I am very active with Dartsch Scientific in this research institute. It's a private research institute which works in different interdisciplinary research in fields of medicine, chemistry, biology, physiology, and biochemistry. And that for more than 22 years. And as a matter of principle, we do not conduct animal experiments, but we work exclusively with organ-specific cell cultures, which we have developed ourselves or developed further. And we work on studies and forecasts on products and their effects on cells. Next slide. First of all, the importance of cell culture in fundamental research and in experimental preclinical research. As the name says it all, we are working with cell cultures we are working with the cultivation of human, animal, and plant cells in an artificial nutrient medium outside of a whole organism. We work with different cell types that can be isolated and cultivated from different organs and tissues. We are working with cells which have a lifespan of limited time, and we've got freshly isolated cells with which we work, and we can work with them in different dishes and recreate the cultures. And this way we have got exactly the same starting experiments, even working with different laboratories. Cell cultures are recognized in the scientific community in the field of medicine, biology, pharmacology, toxology, and biology, and they work as a substitute for animal experiments. So we can investigate substances and effects in order to enlighten the black box. These cell culture studies can be adapted in the experimental design to different test substances and test conditions. That's the huge advantage of a research institute, because we are able to adapt to these conditions and circumstances. If we are working according to standards and norms, that's slightly different in order to work on the biocompatibility. In this case, you have to use standards and norms and subject your test to these. And we have got a bit of leeway with the way we are working. Sometimes you have got tests and studies where you have to look, where you have to adapt certain things in order to get results. And sometimes it takes an awful lot of time. Maybe we can move on to the next slide, please. So what are the most important devices of our research laboratory? On the left hand side, we have got a laminar flow. That's a small device, and in this device you can work with the dishes, with the bottles, and you can open and close them without contamination and fungi. In the middle, a very important device, it's the incubator at 37 degrees Celsius. And we're working with different cells. It depends on the kind of cells, the origin of cells. So you can put dishes and bottles there, and you can achieve a pH at 7.4. On the right-hand side, you see a microscope. The cells are fairly small, you can't see them, and that's why we need a microscope. But this is a special microscope. It is an inverted light microscope. You've got the optics parts below and the light above. So it's inverted light, and there we've got a stage, a kind of table which allows us to work with a big enough distance, so you can handle all the different devices much better than in a normal microscope. So you've got a closer look at the laminar flow. You've got a bottle with culture media, which has got a red color. The red color is an indicator which shows modifications with the change of pH values. So if the color changes, the pH value changes, and you control it at this immediately. Then you've got a pipette sucking up the medium, and you can put that in the various dishes and bottles. This way you can work very effectively. Next slide, please. Again, a couple of pictures from the incubator. On the left-hand side, multiplates. You can have up to 384 holes. And these can be read by computer units. So we are working with 69 whole plates because otherwise it would be too, it would consume too much time and effort. On the right-hand side, you've got culture dishes, 10 centimeters wide, and cell culture bottle with all the red culture medium. we use different water-soluble tetrazolium colors, and we measure the change of colors. So originally the color is red, and because of the medium changes, because of cell activity or free radical activity, the color changes into yellow. Then we've got a second color that changes from orange to yellow. So you've got a 96 hole plate which illustrates that. So at the back there is no reaction, so the color is orange, and the front rows are already yellow, so the cells have already reacted. Then on the left-hand side, you've got a microplate reader or laser reader, which is a photometer where you put these plates inside, and then the optical density is measured and the change of the optical density. And this is measured by splitting the color. Next slide, please. Well, I've already mentioned it at the very beginning. We have measured a bracelet, a wristband, which is called QUANT ARQ. And I would like a little bit about the production qualities. QUANT ARQ is described as a high-end bracelet in high-end quality. It consists of millions of microscopically fine quantum sensors, and these are able to stimulate our cells and increase their vitality. These quantum sensors recognize changes in the cells and send impulses for a better well-being. QUANT ARQ has no expiry date and does not lose its effectiveness. The confirmed effect of QUANT ARQ protects us from undesirable external influences, which are considered to be the cause of oxidative stress, amongst other things. So these are the information by the company, by the manufacturer, and that is also the basis for our tests and experiments. So these are very flowery indications and information, and we wanted to check whether we wanted to check the scientific basis of these statements. We have concentrated our experiments on the biological compatibility, because you wear this bracelet and it's in direct contact with the skin, maybe 12 hours or up to 24 hours a day, just that varies individually. And we wanted to see whether the skin changes. Is this product biocompatible? So that is a first thing that we investigated. And a second point, oxidative stress. Everybody talks about it, and many chronic diseases are based on oxidative stress. So, and the question now is to see whether the QUANT ARQ wristband is able to reduce that oxidative stress, so that stress that comes from the outside, external stress, stress situation, for example. Situations that produces free radicals, which favor oxidative stress. Next slide, please. Well, it's nice to look at this wristband, and that's why I took a photograph to show it. So we use this wristband for our experiments. So next slide, please. I've already talked about the biological compatibility or biocompatibility. What is this all about? In medicine, biocompatible is the term used to describe materials and substances that do not have a negative effect on the metabolism and vitality of living tissues or cells. And now there's one specificity, because Normally, this term is only applicable to medical devices, for example, implants, teeth, or for movement in the musculoskeletal system, which are made of plastic, metals, or ceramics, or also for surgical instruments. There is a standard that describes the exact examination procedure with cell cultures. This is DIN EN ISO 10993-5 from 2009. And they describe the exact examination procedure. Interestingly enough, the biocompatibility test is done with a cell cultures. Normally you use fibroblasts. Here cell lines 3T3 and L929, just by way of example. But Quant ARQ is not a medical product, but we have tested the biological compatibility of this product according to this standard that I've talked about. As I said, when you wear it several hours per day and you're sweating, for example, the sweat might have an effect and a reaction of the skin might be the result. That's why we have done these experiments in order to ensure product quality and user safety. Next slide, please. So the examination procedure, very briefly. The entire wristband was put in a culture medium for 24 hours at 37 degrees Celsius. It was eluated. This eluate was then added to a cell culture of connective tissue cells, and I'm talking here about the cell line L92 in various dilutions. And after 3 days, the cell viability of the cell culture was determined morphologically and enzymatically. And so this procedure allowed the product to be tested in its original state, that is to say, any residues from the production process or foreign substances that could be removed were also evaluated. But let's come to the result. So look at this graphic here, horizontal line, eluate concentration in volume percentage. Medium, only the medium cell medium is at 0, and 900 the cell culture with the eluate. So we've got milli-OD, that is the measure unity in order to measure cell vitality. And we've done studies which go even beyond the standards, for example, with highly dense, high-density cell cultures and lower cell density, so fewer cells in the dishes, and where we added the eluate concentrations. And we looked at the reaction of the cells. But how did they react? Well, nothing at all. They didn't react at all. High cell density, there is no change compared to the 0 value. And the same is true for the low cell density, even if we added the eluate concentration. What does that mean? The wristband is therefore biological biocompatible. That is to say, it was made from the various materials which could cause damage on the cellular level. In all concentrations, the extract had no negative effect on the survivability and the vitality of the test cells. Next slide, please. And you can see that in the cell morphology. On the left-hand side is the control culture without the eluate. So you've got cells which are much lighter. and the others are cells that are separating, propagating. And on the right-hand side, you see cell culture with the eluate, 100% by the QUANT ARQ wristband. There are no cytotoxic effects that can be observed, and that confirms what we have measured beforehand and what I have shown you in the previous slide. So that's on biocompatibility. Now we look at the second subject, oxidative stress. Well, unfortunately, I can't read French, so there has been a slight problem. So I will not read the slide. I will just talk about what you understand by oxidative stress. It's an excessive concentration of reactive oxygen compounds in the body. And this excess can be counteracted by catalase, for example, and cannot be neutralized. That means that oxidative stress represents an excess of oxygen radicals, and not only oxygen, not only oxygen, but also other compounds. These radicals can come from the environment, xenobiotica, for example, environmental pollution, air pollution, or by ionizing radiation or electromagnetic fields, etc. So that's the environment, exogenous effects and radicals. But it's also possible to have a metabolism in stress situation or inflammatory situations in the tissue that can lead to an excess in oxygen radicals, which are formed by the body itself. In the mitochondria, for example, I will show you that in detail later on. When the mitochondria, where the energy is generated in the form of ATP, that's the energy that we need in order to make our body function. In stress situations, for example, it can happen that this metabolism is reinforced, and sometimes oxygen radicals are generated. And through this energy generation, we might have 2 to 3% excess of oxygen radicals. The body's enzymes are capable of neutralizing them in normal situations, but in stress situations we produce much more radicals, and the body defense systems can no longer neutralize them. Too many. And it has been described that this can attack cell membranes. So there might be holes in cell membranes. They can attack enzymes and proteins. And these radicals are also able to attack the DNA and RNA, to change it. And that has genetic consequences. So there are discussions on the aging process, for example, so oxidative in relation to aging and tumor formation. Next slide, please. So I've got This to illustrate what I've just said. On the left-hand side, we've got the endogenous sources, immune system, metabolism, mitochondria, lysosomes, peroxisomes, inflammasomes, etc. On the right-hand side, the exogenous sources, radiation, chemicals, and pathogens. Now we look in the middle. They are the reactive oxygen species. And you've got a number of them who've all got a free electron, and which are very active. So the reactive oxidative oxygen, where that counts also hydrogen peroxide, for example, who hasn't got a free electron, but which has a high oxidative reaction. That is something to remember, because that was part of our experiments on the QUANT ARQ wristband. If we proceed, you've got on the left-hand side antioxidants that fight oxidative stress in order to reduce the number of molecules. We have got secondary plant colors and other substances that count amongst these antioxidants. So negative effects, I've already mentioned it: DNA, RNA, membranes and protein damages, genetic mutations, etc. But there is also the positive side of things, positive effects, for example, of free radicals. They fight killing, they fight pathogens. They kill pathogens. I will talk about how later on. And they're also a signal for the regulation of the metabolism. So we've got negative and positive effects, and oxidative stress is always an excess situation. That is the most important thing. So we've got cellular changes. What do they look like? On the top left, you've got a normal cell culture, fibroblasts, very dark in the middle. You've got nucleus, the cytoplasma. And if you expose this to oxidative stress, by adding peroxide, for example, you see their reaction on the top right picture. So we've got apoptotic cells, so dying cells. Then picture C, so that has increased after 8 hours. So these killing cells have increased in number. And the cells are very weakened, not very much attached to the bottom of the dishes. And D, which is after 12 or 24 hours, depending on the concentration, you clearly see the result. These are only remnants of cells. Everything is more or less dead on this picture. So behind me, I don't know whether you can see that, but there is an inverted light microscope. And maybe, yeah, you can focus on me so that everybody can see that microscope. So you can, well, if you could just enlarge the picture, because then you can have a closer look at the microscope. Well, yeah, the presentation has gone, but maybe maybe you can enlarge my picture. Well, behind me you've got a inverted light microscope, and you see also an incubator chamber, and we have placed a wristband. I don't know whether you can see that. And that is something that I'm preparing for Strasbourg. With this microscope, I can I can film time-lapse situations, and I can show to what extent the quant arc can reduce oxidative stress. So we have got a video where 24 hours can be shown in 30 seconds. That is something that I prepare for Strasbourg. So you are very much part now of the studies. So I have to be very careful as well, because I mustn't touch the microscope, because that will ruin the video. So that's just a small foretaste for Strasbourg. You will be able to see this time-lapse video. Maybe we can go back to this presentation. That would be lovely. Okay, thank you. Can we have the next slide, please? Well, oxidative stress caused by free radicals in the environment. The connective tissue cells, fibroblasts, with which we are working, were subjected to artificial external oxidative stress by the addition of various concentrations of hydrogen peroxide. And this way, it was tested whether the simultaneous presence of the quant arc bracelet had a compensatory effect. And the vitality of the surviving cells was examined after 24 hours of exposure to hydrogen peroxide. And it was tested by quantifying enzyme activity in the mitochondria. The results showed the basically expected concentration-dependent reduction of cell vitality by hydrogen peroxide. So the more hydrogen peroxide, the more cells died. But the vitality values for cells protected by the quant arc wristband were about 20% higher than those of the unprotected cells. That means that the quant arc wristband, under our experimental conditions, had the power to fight off these free radicals that were in the hydrogen peroxide. So there was a compensation procedure, 20% high. If that had not been the case, then we couldn't go on with the time-lapse videos, for example. So that is not the first video that we are registering. But we have seen that the quant arc wristband had a positive effect and protected against the hydrogen peroxide that we had added. So oxidative stress through endogenous radicals, that is to say mitochondria. On the left-hand side, you've got a cell, you've got the nucleus and other elements, endoplasmatic reticulum. We are interested in the mitochondria. These are structures that we have isolated. Here on the right-hand side, you've got a picture. They've got exterior membrane, interior membrane, and they change oxygen, carbohydrates, lipids, etc. That's cell respiration, so to speak. And when we are in a stress situation, then there is more production of energy, so are we sort of fiddly, and our vitality status has changed. And in these situations, more oxygen radicals are being produced. Because in this process, oxygen radicals are being produced. Normally, they are dealt with by our body, but in case of excess radicals, this is no longer possible. Then we have got another oxidative stress by neutrophil granulocytes. This is an innate immune defense, but they can also migrate into the tissue and cause production of oxygen radicals, and that can lead to undesirable tissue destruction. And that can lead to inflammatory processes and can codify these. So these cells have a positive effect. So they defend the body against pathogens. They can neutralize them, they enclose them, so to speak, in form of an oxidative burst. And this way, the pathogen is killed and destroyed. And this pathogen that had been killed will then be sort of eaten up by these phagocytes. That's their function in the blood. And the other function is not very positive, as I've already explained. We can generate a functional neutrophil granulocyte. We work with a cell line HL60, and if you add DMSO after 5 to 6 days, you can differentiate them in order to generate functional neutrophil granulocyte. And stimulated by a Fobol ester, they can generate cyperoxidanium radicals. And this way you can test the effects. You can test the effectiveness via a test assay measuring change in color in relation to the number of radicals that had been generated. Next slide, please. So I've illustrated an oxidative burst. On the left-hand side, you see oxygen plus water and the reaction together with the enzyme that generates a superoxide, that's O2 minus, and the lonely electron is missing. So this is a reactive oxygen species which can be generated. On the right-hand side, you see a picture of a neutrophil granulocyte. Next slide, please. There have been various studies on the QUANT ARQ wristband in order to test to what extent it is capable of combating the oxidative stress. On the left-hand side, you see the control, and on the right-hand side, the situation with the QUANT ARQ wristband. The green shows the basal metabolism, that is to say normal metabolism without a stimulation. The red shows the cell metabolism and the activation of the radicals and the radical formation, so you can see the change in color. So in both cases, The bar on the right hand, the bar is higher. But on the next slide, you can see something astonishing. So the part of the radical generation in form of oxidative burst. I've changed the color. On the left-hand side, the oxidative burst is much higher, so more radicals had been formed without the quant arc. On the right-hand side, the yellow field is much smaller. So 75% fewer radical formation thanks to this quant arc wristband. That is really astonishing. Next slide, please. Well, let me come to the summary of my presentation. The results of the studies show the great potential of the quant arc wristband to reduce the effect of an excess of radicals, the oxidative stress. And it makes no difference whether the reactive radicals originate from the environment of the cells or are formed by the cells themselves. And furthermore, the quant arc wristband and its materials used have been tested as biocompatible. And that, as you can see on the next slide, that meant that we have a certificate that is available in different languages, and that certificate summarizes what I have shown you tonight. Thank you very much for your attention. I'm always pleased to come and talk to you about our latest research results. Thank you ever so much. Thank you very much for this presentation. Excellent presentation, because we witnessed the effects of this product, and I'm really pleased that you will present us with further news in Strasbourg. Those who haven't organized their trip to Strasbourg, please do so and get your tickets. Professor Dr. Dartsch, again, thank you ever so much for being with us today. Each time we get a very positive feedback for your trainings, and our customer service will send you some questions, because if we'd accept questions now, we will stay all night. So we will select the questions and will forward them to you by email. Well, yes, as I said, I'm always very pleased to talk to you and talk about the latest research results. I'm very pleased to see you again in Strasbourg, and you, ladies and gentlemen, you will have the possibility to see me in real life, not only in front of a computer. And if it's possible, you can also talk to me and ask me questions, but let me eat from time to time. Otherwise, I'll be at your disposal. Thank you ever so much again, and have a nice evening, everybody. Bye-bye.